Hello, everyone, and welcome to the fourth week in our online Evolution and Ecology seminar series. My name is Generality, and I'm a postdoc in Judith Mang's lab at the University of British Columbia. Myself and the other organizers, Elizabeth, Andreas, and Wouter, are thrilled to be hosting some of the most exciting evolution and ecology scientists to share their research. Before I introduce today's speaker, I just want to quickly remind you of our Q&A session. If you would like to post questions for the speaker, you can do so in our designated Slack channel, where you can also upvote the questions that you would like to see answered. I would also like to take this opportunity to thank you all for watching the seminars, for posting all those great questions, and just for your continued support in this. So thank you, and please do continue to spread the word about these great seminars. And with that, it is my pleasure to introduce today's speaker, Professor Chelsea Wood from the School of Aquatic and Fishery Sciences at the University of Washington in Seattle. Chelsea is an expert in disease ecology, studying how parasites and pathogens in marine and freshwater ecosystems respond to human impacts on the environment. Chelsea's work also focuses on using ecological techniques to develop much-needed solutions for infectious diseases. And today, she's going to tell us more about what can data on historical parasite burdens reveal about the future of marine diseases. So thank you very much, Chelsea, for accepting our invitation, and over to you.
Thanks, Julia, and thank you to all of you watching for having me. I'm Chelsea Wood. As Julia mentioned, I'm a parasite ecologist at the University of Washington in Seattle, and I was so pleased to get this invitation. Like many of you, I have been holed up in my apartment since March, putting my ecology class online and plugging away at analysis and writing projects. And like many of you, in those hours not spent panicking about online teaching, I've been watching TV and revisiting some favorite movies. Because I have an extremely serious case of cabin fever, I hope you'll indulge me just a little bit. I want to show you my vision for the future of marine disease. It's going to involve looking to the past, and along the way, I'll show you some recently published and some unpublished data, and we'll throw in just a little bit of Spielbergian magic. Today, I'm going to tell you about data that came from a huge field and lab effort. So I want to make sure that you know upfront that I didn't do this all alone. This work is the result of joint efforts with many collaborators at UCSB, as well as Scripps, Stanford, Washington Department of Fish and Wildlife, and of course, UW. And it would not have been possible without the generous support of lots of funding agencies. One of my all-time favorite movies is Steven Spielberg's 1993 Jurassic Park, which just had its 25th anniversary. One of the things that I love about this movie is the way that it shows what it feels like to be a scientist who studies the past. You make the inferences that you can make with the scraps of information that are left to you in the present day, but you yearn to see those past ecosystems with your own eyes. Let's set the scene. Dr. Alan Grant and Dr. Ellie Sattler are just entering Jurassic Park for the first time with no idea of what the park is or what it's meant to contain. They have these theories about dinosaur behavior, but they're just untestable theories. They're based on biomechanics or on the behavior of the dinosaurs' descendants, the modern day birds. They don't think they'll ever have the opportunity to test these theories in real life because they don't think that they'll ever have the opportunity to observe dinosaur behavior in real life. And then these scientists find themselves transported through time. Their lives were spent studying the remnants, the artifacts of this lost world. And then this world is found again. Today, I'll show you my vision for the future of marine disease and how new approaches are allowing us to do what Doctors Grant and Sattler did. I'll tell you the story of how I went from using the behavior of modern day birds to make inferences about the behavior of dinosaurs to actually watching dinosaurs behave in real time. Metaphorically, of course, because you guys didn't bring me here to talk about dinosaurs. What I study is far more beautiful and far more dangerous. I'm interested in marine infectious disease and the parasites that cause those diseases. And today I'll tell you the story of how I went from using the scraps of information that we could glean about the dynamics of marine infectious disease from contemporary ecosystems to literally watching parasite populations change across time. I'll begin with the theoretical background that motivates my work. I'll then briefly walk you through our work in contemporary ecosystems where we're using space for time substitution to try and understand how various human impacts change the abundance of marine parasites. But I want to spend the bulk of my time today talking to you about brand new, unpublished efforts that take us from contemporary ecosystems deep into the past. And then I'll wrap up by drawing some conclusions about what marine disease of the past has to teach us about marine disease of the future. Let's begin at the beginning. I started in on this line of research because there's one question that really drives my interest in marine biology, and it is, what did the oceans look like before people? The world's oceans are being remade by a growing human population. If you spend time on the water, you probably have some intuition that marine ecosystems look a lot different today than they did in the past. And that intuition is not just nostalgia. There are hard data to suggest that the oceans have changed substantially in the past decades, and that fishing is primarily to blame for this. So we were talking about how the oceans have changed substantially and how fishing is driving that change. And I was showing you these pictures from a 2009 paper that Lauren McClenahan wrote. And she gathered up these pictures from a public library in Key West, where she sort of stumbled over a shoebox of these old trophy photos. And they show the same hanging board on the same wharf over the course of 60 years. And the photos reflect and document the state of Key West reefs over this long time period. There are a couple patterns that we can pick out in these pictures. Over time, fish become fewer, they become smaller, and their average trophic level declines. And this is just one particularly visual example, but of course, this is not a phenomenon that's limited to Key West, it's a global phenomenon. So historical work gives us a pretty good idea of what the oceans used to look like before these impacts accumulated. We have decent data for many of these large charismatic species, but as many of you are aware, my interest in fish goes one level deeper. Each fish here is an ecosystem in itself, and that internal ecosystem is typically concealed. But what if we could unveil it? What sorts of parasites would we see in this scene? Were there more parasites in an unfished ocean, or fewer, or the same overall number, but a different mix of species? We have no idea. Now, some of you in the audience might be thinking to yourselves right now, who cares, right? Who cares how many parasites there were before fishing? They're small, they're unimportant. We've got much bigger things to worry about, like the loss of all these beautiful charismatic vertebrates. But parasites have big impacts on the rest of biodiversity. And I want to give you just one example. This guy who's wiggling in the upper left is an anisakid nematode, and it's occasionally passed to humans when they consume uncooked fish. I am not going to gross you out with the symptoms, but suffice it to say they are not fun. And the same goes for the effects of all of these parasites on their wildlife hosts. So if fishing does cause changes in parasite abundance, that could end up being really important for these vertebrate animals that we care a lot about. So here we have a big unanswered question in marine biology. And one of the things that makes this question really interesting is that we can make reasonable hypotheses that predict very different outcomes. It's easy to imagine how fishing might increase or decrease parasite abundance depending on its effects on fish hosts. So here I'm going to use the same time series of dayboat trophies in Key West. I'll layer on some dots to represent fish parasites. And this conceptual diagram is going to illustrate what's happening in the photos. Fishing pressure increases as we move through time and across the x-axis. Now, fishing doesn't reduce the density of every single fish species, right? Fishermen like to catch big, high trophic level species like sharks, but that's good news for the lower trophic level species that get eaten by sharks because they could experience compensatory increases in their abundance. And if that lower trophic level host is increasing in density, we might expect to see a matching increase in the abundance of its parasites. In the disease ecology literature, this would be called the dilution effect hypothesis. But it's also easy to imagine how we might see the exact opposite pattern. Hosts are both habitat and resources for their parasites, and parasites could become less abundant as fishing proceeds because fishing reduces fish density or size or the trophic complexity of fish assemblages, all of which should affect parasites. I'm going to call this the healthy ecosystems are rich in parasites hypothesis after a tree paper by Pete Hudson, Andy Dobson, and Kevin Lafferty. But of course, when I say the word parasite, I'm invoking this sprawling group of organisms that are diverse taxonomically, morphologically, and trophically, and there's no reason to expect them all to do the same thing. So we might expect a mixed response of parasite abundance to fishing. Here I've changed the colors of the dots just to indicate what line they correspond to in the conceptual diagram. And that begs a big and really important question, which is how does fishing affect whole parasite assemblages? Which parasites win, consistent with the dilution effect? Are there any that lose, consistent with the healthy ecosystems are rich in parasites hypothesis? And can we use some information to predict those outcomes? Years ago, lacking the ability to peer back in time, I set out to answer this question by using space for time substitution. And this is basically the approach where we find contemporary ecosystems that look the way that ecosystems looked in their pristine condition. And in that way, a pristine ecosystem that exists today stands in for a pristine ecosystem of the past. We worked in the Northern Line Islands. This archipelago contains both uninhabited and unfished islands, as well as other islands that are inhabited and fished. This box is going to go around Hawaii. About 1,000 miles south of the Hawaiian archipelago are the Northern Line Islands. This is a color code that I'll use throughout the talk. Blue is going to indicate unfished islands, and brown will indicate fished islands. And you can see that we've got 3 fished islands in the middle and 3 unfished islands on either end. This archipelago spans a gradient in latitude, and so in the associated biotic and abiotic conditions that are correlated with latitude. But because of that mixed distribution of fished and unfished islands, we can parse those potential confounders from fishing effects. The fished islands are Turena, Tabuaran, and Christmas. They're part of the Republic of Kiribati, and each is inhabited by between 1,000 and 8,000 Kiribati people. Most of those folks rely on subsistence fishing. And you wouldn't expect such low fishing pressure from such a small human population to have a really strong effect on the fish community, but it actually does. We see very few of the top predators that are favored by fishermen on these fished islands. Contrast that with our unfished islands. Jarvis, Kingman, and Palmyra are owned by the United States, and they're managed as part of the US Pacific Remote Islands Marine National Monument. And that designation involves really strict bans on fishing that are well enforced. As a result of that protection, these islands are just teeming with predators. Sharks, jacks, snappers, groupers, you name it, it's out there. So these islands serve as a window into the past. This is what most coral reef ecosystems used to look like. So these islands are a living laboratory for understanding how coral reef ecosystems used to function. We set out to understand how fishing drives change in parasite assemblages by conducting sampling across those 6 islands. The sampling was done on a research cruise that was conducted in the fall of 2010. It was led by my collaborator, Stuart Sandin from Scripps, as well as his graduate student, now postdoc, Brian Zaglinski. They sailed from Oahu all the way down the Line Islands chain, and at each of those islands, they collected fish with pole spears. We focused on 7 host species, and these were targeted to span a variety of trophic levels and body sizes so that we would maximize the taxonomic diversity of parasites that we would detect. Ultimately, we dissected 945 fish for the project across 7 host species and 6 islands for a total of 44 parasite species, just shy of 1 million individual parasites. And that might sound like a lot, but it's actually a pretty average burden for tropical fish from this part of the world. For each of those fish, we examined every organ individually for metazoan parasites. We didn't look for protozoa, viruses, or bacteria. And before we jump into the results, let me get you oriented to one panel of what will ultimately be a multi-panel plot. This one applies to Tinochaetus marginatus, our bristletooth. On the y-axis, you'll find all of the parasite taxa that we detected in Tinochaetus marginatus. On the x-axis is a standardized regression coefficient for the effect of fishing. So this is a coefficient that comes out of the generalized linear mixed model that you see described at the bottom of the slide. And basically this x-axis indicates the response of the parasite to fishing pressure. So positive values on that x-axis indicate a positive response to fishing pressure, or parasites where we found more individuals on fished than on unfished islands. And then negative values on that axis indicate a negative response to fishing, or parasites where we found more individuals on unfished than on fished islands. Because this is a standardized regression coefficient, anything greater than +2 or less than -2 is significant, and that's why those 2 areas are shaded. And all the p-values that you're going to see have been corrected for multiple comparisons. So with all that in mind, let's take a step back and appreciate that what this plot is showing us is that there is a whole mixed bag of responses among parasites. We've got a bunch that fall in the middle that don't have a systematic response to fishing pressure. They're equally abundant on fished and unfished islands. And then we've got 3 parasites that fall into that brown zone on the right who are conforming to the predictions of the dilution effect. They're more abundant on fish than on unfished islands. And we have 3 parasites on the other side that conform to the predictions of the healthy ecosystems are rich in parasites hypothesis. They're more abundant on unfished than on fished islands. If we then zoom out and take a look at the whole plot, we see that that same pattern holds. A totally mixed bag with many parasites not responding, some responding positively and some responding negatively. Now, the fact that there are such mixed responses from parasite species to parasite species is in and of itself a really interesting result. Dilution doesn't happen in the majority of cases. And in fact, we often see the very opposite of dilution, parasites disappearing as fishing pressure increases. So these data suggest that we can't expect a homogeneous response to fishing across our parasites. We could have stopped there. But we had some hypotheses about why there might be this kind of variability, and we thought that it might be related to parasite traits. Let's start peeling back the layers of this onion a little bit. We sorted our parasites into two broad transmission strategies: directly transmitted and trophically transmitted. Directly transmitted parasites are passed from one host to another host of the same species, but trophically transmitted parasites use multiple hosts of different species. We sometimes also call them complex life cycle parasites. We found parasites in both of these groups. Among our directly transmitted parasites were the crustaceans and the monogenes. Among our trophically transmitted parasites, nematodes, cestodes, trematodes, and acanthocephalans. And we made some predictions based on these fundamental life history differences between parasites. We assumed that these parasites would be transmitted in a density-dependent manner, so that if host declined in the presence of fishing, parasite transmission would decrease and parasite abundance would decrease. Basically, the equivalent of social distancing, right? If we reduce host density, we reduce transmission. But we also know that this isn't the only response that fish populations can have to fishing. If fishing removed the predator of this host, we might see a corresponding increase in host density. And if the host is increasing in density, we would expect its parasites to profit alongside it. This is the equivalent of beaches opening up and people sitting blanket to blanket in the middle of a global pandemic. So our hypothesis was that the directly transmitted parasites would track their hosts. In contrast, the trophically transmitted parasites are dependent on multiple host species for the completion of their life cycles, and those life cycles often include the large, high trophic level species that are favored by fishermen, like sharks and big predatory teleosts. And we know that if you lose just one link in this parasite life cycle, it's game over, transmission stops. So we expected trophically transmitted parasites to be especially susceptible to the impacts of fishing on their hosts. To test these hypotheses, we wanted to turn this analysis on its head. We essentially wanted to group up these regression coefficients by parasite type and find out whether there were any patterns. Now remember, this is a space for time substitution. We want to get some hints about what parasites would increase and which parasites would decrease in a fished ocean. So to answer that question, we asked whether the mean regression coefficient within a bunch of different parasite taxa differed significantly from zero. And we call that the mean effect size. In this plot, we broke down that mean effect size within a bunch of different higher order parasite taxa. So that's why you see crustacea, monogenes, trematodes, cestodes, nematodes, and acanthocephalans on the y-axis. On the x-axis is that mean effect size, and just like before, brown is a positive response to fishing, where we find more parasites on fished islands in responses that are consistent with the dilution effect hypothesis. And then negative values indicate a negative response to fishing, where we find more parasites on unfished islands, consistent with the healthy ecosystems are rich in parasites hypothesis. I'm going to plot these mean effect sizes with 95% confidence intervals, so remember that anything that overlaps is not significantly different. If we begin at the top, we see that the crustaceans and the monogenes both have significantly positive responses to fishing. They're more abundant on fished than on unfished islands. And if we combine those together, there are two groups of directly transmitted parasites. We also get a significant positive response. So this is a little bit different from our prediction, right? We thought that the directly transmitted parasites would track their hosts. Why are they all increasing? Well, it turns out they're doing exactly what we predicted. The majority of the fish that we had in this study actually increased in response to fishing pressure. And it's the parasites of those fish that are driving the positive response here. So that's what's going on with the directly transmitted parasites up top. If we move down to the trophically transmitted parasites, it's a different story. The cestodes and the nematodes each have significant negative responses to fishing. They're more abundant on unfished than on fished islands. And we only found this one acanthocephalan, so it was not included in the meta-analysis, but I wanted you to see that its regression coefficient was non-significantly negative. And so far, this is all consistent with our hypothesis that fishing should interrupt the life cycles of trophically transmitted parasites and cause them to decline in the presence of fishing. But the trematodes do not follow this pattern. Their response to fishing doesn't differ significantly from zero, and that's a little bit of a departure from our hypothesis and from the findings for the remainder of the trophically transmitted parasites. We'll talk in just a couple seconds about why I think this might be, but if we average over all the trophically transmitted parasites, we see that their response doesn't differ significantly from zero, but it is significantly less positive than the response of the directly transmitted parasites. Okay, what's going on with the trematodes? I think it has something to do with their life cycles. The life cycles of the cestodes, nematodes, and acanthocephalans differ fundamentally from the life cycles of the trematodes. If you are lucky enough to begin your life as a cestode, nematode, or acanthophyllin, you are born in the intestine of a large predatory vertebrate. You're then pooped out into the water column, where you hatch and infect a first intermediate host crustacean like the planktonic copepod shown here. Trematodes also use large predatory final hosts, but they prefer teleosts to elasmobranchs. And their first intermediate hosts are not planktonic crustaceans, but instead are benthic snails. Now, planktonic crustaceans are pretty insensitive to fishing impacts. Their abundance is determined by bottom-up forces like oceanic productivity. Fishing generally doesn't affect their abundance. So when a nematode, tapeworm, or acanthophyllin life cycle is hit by fishing, the effect is straightforward, right? We get rid of that final host and transmission is reduced. The effects of fishing on trematodes might be a little bit more complicated because while their final hosts certainly are the targets of fisheries, fishing has effects on other parts of their life cycle as well, because snails are major prey of fish species. And we know from work on Kenyan coral reefs that they can sometimes increase in abundance in fished areas where they escape their predators. So trematodes might get these counteractive effects, reductions in transmission due to fishing-driven loss of their final host, but also increases in transmission due to fishing-driven compensatory increases in the first intermediate host. And if you think about it, it's kind of a fundamental question in parasite ecology, right? Like, where is the rate limiting step in this life cycle? I don't know the answer to that question, but I do want to give you some insight into why I offer this explanation for the equivocal response of the trematodes. And it has to do with this one weird outlier trematode. Stephanostomum is a trematode metacercaria, a larval trematode that we found in 5 different host species. And it had this really positive response to fishing pressure. Like I said before, trematodes usually use snails as their first intermediate host, but Stephanostomum uses a very particular type of snail. Big, meaty moon snails and whelks, the snack food of the reef. Snails that are especially susceptible to predation pressure. So while Stephanostomum's definitive host, final host, might be fish, it could be experiencing a strong increase in the abundance of its first intermediate host, which might explain why it has such a positive response to fishing. So snails mess this up. They make it way more complicated. And if we consider only the trophically transmitted parasites that lack snail intermediate hosts, they do have a significantly negative response to fishing pressure. And altogether, what these data tell us is that there are both winners and losers among parasites in a fished ocean. For our system, the losers include the trophically transmitted parasites, especially the cestodes and the nematodes. The winners are directly transmitted parasites of hosts that increase with fishing, and trematodes with intermediate hosts that increase with fishing. Now, we were really excited by this finding. This suggests that the oceans might have looked a lot different in the past than they do today. Not necessarily that they've undergone an overall increase in disease or a decrease, but a shift in the kinds of parasites that are common. I could go on for another 2 hours, giving you all the evidence that we've amassed to suggest that this pattern holds up in other ecosystems. And if you're interested in seeing those data, please let me know in the Q&A. I'll point you in the right direction. Suffice it to say, we've done additional studies ourselves in my lab, and we've meta-analyzed the studies of other folks, and this pattern appears to be robust across marine ecosystems. So we find these very robust patterns of fishing driving increases in directly transmitted parasites and declines in trophically transmitted parasites. And that's all well and good, but contemporary ecosystems are not a perfect stand-in for past ecosystems. I'm going to skip quickly past this so that we don't get bumped off again. But the point that I was trying to make in that little clip that is clearly a copyright violation is that contemporary ecosystems are just a pale imitation of historical ecosystems. If we really want to know how parasite assemblages have changed over time, we need to find some way to get data on parasites of the past. We wanted to take the hypotheses that we had generated in our work on contemporary coral reefs and see if they could predict how parasites have actually changed over time. And so let us now pass through the gates of Jurassic Park, literally stepping back in time. So people have come up with some really creative ways to glean information about historical assemblages of fishes. Some have used art, like this Roman mosaic showing a dusky grouper, a species that's now endangered and very rare in the Mediterranean and even more rarely eats people. Others have used the composition of species represented in seafood menus to learn about change in fish assemblages over time. And of course, people like to take pictures of the big fish that they catch, and creative folks like Lauren McClenahan have extracted from these kinds of photos qualitative and sometimes even quantitative data to contrast against modern fish assemblages. And these pictures are just, they're really striking. They bring home the fact that the oceans look very different today than they did even just a couple of decades ago. Now, parasites are just not that charismatic. No one takes pictures of them while they're on vacation. You're not going to find them on a menu, and not many works of art are devoted to them. And so historical information on parasite assemblages is harder to come by, except for one source: museum specimens. When fish or invertebrates are preserved in ethanol or formalin, their parasites are preserved right alongside them. We're lucky at UW to be home of the Burke Museum and its ichthyology collection, which is the largest collection of North American fishes with 11 million catalog specimens, and some collections dating from the early 1900s. And that sets us up to be able to reconstruct historical parasite assemblages over a really broad sweep of time. So we'll return to the Burke in just a few minutes, but first let me set the scene for this next project that I want to tell you about. My adopted hometown of Seattle is a fishing town, and it has been for a long time. Today, it's the home of the US North Pacific Fishing Fleet, which ranges over thousands and thousands of miles of open ocean. But in earlier days, boats based in Seattle focused primarily on Puget Sound. Here's Puget Sound, an ecosystem that will be familiar to many of you, located in the US Pacific Northwest. These dots represent the contemporary density of human population across the landscape, and Seattle is in that lower right-hand corner. Today, not a lot of ground fishing happens in the US part of the Sound, but for much of its history, trawling was the most important part of the Washington state fishing industry, and one of the biggest earners were English sole, which produce these lovely, mildly flavored fillets. Now, you guys, you know that this is a parasite talk, so any appetizing slide is going to be immediately followed by an extremely unappetizing one. Around the 1930s, folks started complaining about these things showing up in their delicious sole fillets. By the late 1940s, infections were bad enough that tons of English sole that were landed in Seattle needed to be diverted to animal feed. They actually went to mink farms. They were too wormy for human consumption. This is Clavonema mariae. It's a nematode bloodworm parasite that's closely related to a worm that humans get called guinea worm. The brand new Washington Department of Fish and Wildlife did its thing. They commissioned a research cruise to investigate the distribution of this parasite, hoping to find some part of Puget Sound that could still be profitably exploited. So they hired the RV Panther. The Panther went out in 1949 and 1951. They collected reams and reams of data on the abundance of Clavonema mariae in English sole. But they failed. There was apparently no place in the southern part of Puget Sound that was free of this parasite. Their data got filed into a cardboard box and dumped into the basement of WDFW headquarters in Olympia, Washington. When I first started at UW 4 years ago, I got a call about this original data set from a WDFW scientist who had been rooting around in that basement. I had been searching for years for good historical data documenting marine parasite abundance for any parasite in any ocean at any historical time point. I can tell you these data are exceedingly rare. And so this Clavonema mariae dataset fell into my lap like a gift from God. Another gift from God arrived shortly after. Ingrid Howard, an amazing undergraduate who graduated 2 years ago and incidentally is looking for a job. So if you have the opportunity, I encourage you to hire her. All the data that I'm about to show you come from Ingrid's capstone or undergraduate honors thesis project, which was recently published in the Journal of Applied Ecology. If you're interested, I encourage you to check out the details there. Okay, so Ingrid started by looking at all the historical data that were available. We entered all the data from the WDFW commissioned Panther cruises in 1949 and 1951. And because this is an economically important parasite, we were also able to gather data from some literature resources. We then recreated all of these historical sampling efforts in 2017. We sampled the same locations using the same gear at the same depths. And here on the x-axis, I'll show you 5 locations where we were able to obtain both historical and contemporary data. On the y-axis, you're going to find the prevalence of the parasite Clavonema mariae. For every site, historical prevalence was lower than contemporary prevalence, and on average, prevalence was almost 50% lower in the past than at present. So that's super interesting. But this is one of those rare economically important parasites where we actually can find historical data. And I'm curious about all the parasites, not just the handful that we happen to have data for. We wanted to see whether we would get the same story if we looked to the museum specimens. And this would serve a couple purposes. First, it would let us confirm this really interesting pattern because we would be testing for a temporal pattern with 2 completely orthogonal data sets. And if we got the same story, it would bode well for all those other parasites for which we have no historical data to use for ground truthing, kind of like a proof of concept, testing whether you get the same answer from looking to the museum specimens that you do when you go out and actually measure the parasite in nature. After all, the museum specimens could be a biased subset of what was available at the time of their collection. But if the historical data and the museum specimens do tell the same story, that means that they might be a reliable way to reconstruct parasite assemblages of the past, and we might be able to use the museum specimens to reconstruct change over time for those parasites where there is no historical record. So Ingrid set out into the UW ichthyology collection, and she pulled every single English sole collected from Puget Sound. She counted the number of Clavinae mammariae in each of these and recorded the year of their collection, and this is what her data looked like. Each fish had on average 0.11 worms at the beginning of the data set and 0.86 worms at the end of the data set, a nearly 8-fold increase in burden. So we get the same story, whether we look at the historical record or the museum specimens, and it's a story of increasing parasite abundance over time. But of course, that's the story of only one parasite in one host. And we wanted to be able to tell a much grander tale across many parasite species. So I launched a new project that my lab likes to call Parasites of the Past. The project is supported by the UW Innovation Imperative and the Alfred P. Sloan Foundation. And it's being carried out in collaboration with Luke Tornabene, the curator of fishes at the Burke, and Catherine Myslenikov, the collections manager there. We're expanding to look at all the parasites in museum specimens of 10 Puget Sound fish species. And these species, again, were chosen to span a variety of trophic levels and body sizes, again, so that we would maximize the diversity of parasites that we would find in them. And most of them are available right here in the UW fish collection, although we'll be supplementing from other collections as well. And we should be able to get back into the 1880s for many of these species. So we just looked in English sole, we saw that this one parasite exploded in abundance over time, and we wanted to know, would this be the story for all the parasites in all of these hosts? I can't answer that question for you yet, but I do want to tell you about the first bits of data that are coming out of this project. We wanted to know, do the predictions we generated in our work on contemporary coral reefs jive with how parasite assemblages have actually changed over time? With the idea being, we want to look at trajectories of parasite change over a century-long timescale. But first, we needed to answer another really pressing question. Are museum specimens reliable? Can we count on them for accurate information about how many parasites were in a fish at the time of its death? Ingrid's study showed us that we get the same direction and magnitude of change over time, whether we look at the museum specimens or the historical data. But it still might be possible that it's just tougher to find parasites in a fixed fish specimen. And so museum specimens could consistently underestimate the true abundance of parasites for any given fish. And so we set out to answer this question. And when I say we, I mean my master's student, Evan Fiorenza, who graduated last year. Evan got a hold of a bunch of fish and he randomly assigned them either to be preserved with the preservation protocol that the UW Burke Museum ichthyology collection uses to fix their fish, or they were put in a control group where nothing was done to them. And then he performed parasitological dissections and looked for differences between those treatments. What Evan found was that there was a minimal influence of preservation on parasite detectability. He found 27 parasite species, and of these, only 3 differed between the control and the preservation treatments. From here on out, I'm going to ask that you refrain from taking pictures of the slides or live tweeting the talk, because the entire rest of this talk consists of in review, in revision, or in prep work, some recently accepted. But I do hope that you enjoy getting a sneak preview of the latest stuff coming out of our lab. Okay, so Evan aggregated the response to preservation across all of the 27 parasite species detected in these 3 hosts that he examined. And he found that no group was easier to detect in the preservation treatment, oops, sorry about that, versus the control treatment. So values on the right of this axis indicate parasites that were easier to detect in the preservation treatment, and values on the left of this axis indicate parasites that were easier to detect in the control treatment. These are 95% confidence intervals. So you can see, no matter how you slice it, there's no effective preservation on detectability, whether you look across all taxa stages, whether you separate the larval parasites from the adult parasites, and whether you look at the taxonomic groups of parasites separately. Okay, so between Ingrid's study and Evan's, we can be pretty confident that the museum specimens are providing us with reliable information on parasite abundance. And with that in mind, we set out to answer our core research question: how has parasite abundance changed over the past century? We've assembled a crack team to address this question: Rachel Wellicke, Whitney Presser, Kitty Leslie, and Natalie Mastick. The work that I'm about to show you is led by Rachel, and she's the first author on the resulting publication, which was just submitted last week. Now, these guys are dissecting our 10 selected host species, and these are literally parasite time capsules passed down to us from decades ago. We're just finishing the first year of this project. We've finished dissecting 4 host species, and we finished the data cleaning for one of those species, the one shown here, English sole. The data cleaning takes some time because we're confirming all of our taxonomic IDs with Mike Kinsella, who is retired but graciously working with us. So 3 species are in the early stages of data cleaning, but I do want to show you the results for our very first host species of the project. I'll begin just by getting you oriented to one panel of what will be a multi-panel plot. What I'm going to show you are a bunch of time series spanning from about 1930 to 2016. On the x-axis will be year, and on the y-axis, parasite abundance. These are actually partial residuals that adjust for differences in fish body size across time. We're going to throw a regression line through each cloud of points with an associated 95% confidence interval, and each plot will represent a single parasite species. I'll indicate to you the taxonomic group of the parasite with these cartoons. We'll have copepods and leeches among our directly transmitted parasites, and trematodes, nematodes, cestodes, and acanthocephalans among our trophically transmitted parasites. And I've grouped up all these parasites by their responses. In total, we detected 12 parasite taxa in this one host species, English sole. Over the 85 years of the data set, 9 of those 12 parasite taxa have not changed in abundance. Notice that there are a couple of directly transmitted parasites here, leeches and copepods, also a couple of trophically transmitted parasites. So that's 9 out of 12 that do not change over time. We have 1 out of the 12 species that increases in abundance over time. This is a trematode larva called a metacercaria that we detected far more of in recent years than we did in the past. And then finally, there were 2 species that have actually declined through time. Here we have 1 acanthocephalan and 1 trematode, both trophically transmitted parasites. But these are just the results for individual parasite species. What if we harness replication across parasite species and ask about trends for different parasite groups? Here are our results summarized across parasite groups. Positive values on the x-axis indicate parasite taxa that have increased in abundance through time, and negative values indicate parasite taxa that have declined through time. Neither directly transmitted parasites nor complex life cycle or trophically transmitted parasites have a consistent direction of change in the aggregate. The one place where we do see a significant effect is among the acanthocephalans, a group of trophically transmitted parasites or complex life cycle parasites, which have clearly declined through time. So this doesn't strictly match our prediction that directly transmitted parasites would increase over time while trophically transmitted parasites declined. But as many parasites are declining as they're increasing, and the trophically transmitted parasites are tending to be the ones who decline over time. So I don't know, they're sort of moving in herds. I'm not nearly as prescient as Alan Grant. We have a lot more work to do on this project, 9 more host species coming up, and we're hoping that we will end up seeing some clearer patterns as we increase replication across parasite species. But the big question that we face here is why? What are the factors driving the change in parasite burden through time? Because fishing isn't the only thing changing in this ecosystem. Maybe the changes that we've observed in the parasites of Puget Sound are driven by this steady incremental increase in sea surface temperature that we see year after year, which could speed up parasite life cycles. Or maybe it's driven by the change in the burden of nutrient pollution due to development, agriculture, logging. That could facilitate blooms of the intermediate host copepods needed by many of these parasites. Or maybe it's due to the ebb and flow of host density over these 100 years, because we know that English sole hasn't experienced a steady drumbeat of increasing fishing pressure. It was fished really heavily for a long period of time, and then fishing pressure relaxed. Any of these explanations are plausible, but which ones actually correlate with change over time in parasite burden? Well, we're super lucky because these major drivers are relatively independent and uncorrelated with one another, and because Puget Sound is a well-studied region and has some really good historical data on each of these potential drivers. There's a continuous record of sea surface temperature from 1921 to the present day from Race Rock's lighthouse in the Strait of Juan de Fuca. There's a great study documenting biomarkers of nutrient pollution in sediment cores covering the period of 1880 to 2000. And finally, there are historical fisheries data documenting the relative density of most of the hosts that we're interested in. Again, we are super lucky to be just a few steps from some folks who've already been hard at work reconstructing the density of these fish populations. We're working with Tim Essington in my department at UW, as well as Jamil Samhori and Corey Green at the Northwest Fisheries Science Center to chase this down. And in the next few years, we hope to have some hints about the forces that are correlated with parasite increase and parasite decline through time. So while I can't answer this why question for you, rest assured we are getting closer, both to describing the patterns of parasite change through time and to narrowing in on some of the potential drivers of that change. The past is past. We stand at a moment of unprecedented change for Earth's ecosystems. So what does all of this mean for the future of marine disease? What's going to go on under the surface of the visible world among all these cryptic hidden parasites that are typically unseen? Are we in for an explosion of marine disease, or will future oceans see the loss of parasite species, even those that contribute to ecosystem function? I can't say with a ton of certainty at this point which are the parasite taxa that are likely to increase and which are the ones that are likely to decline. But I do know that we will see both of those things happening as we move into the future. We're going to lose some parasite species and we're going to gain others. And both of those outcomes have important implications for our ability to feed the world with seafood and rely on functioning coastal zones for ecosystem services. The losers probably include lots of parasites with important roles to play. And that might sound counterintuitive, right? Parasites as a good thing in the ecosystem. But parasites are natural parts of ecosystems. They're species organized into populations, and they perform vital ecosystem functions, especially the trophically transmitted parasites. We can talk more about this in the Q&A if you're interested, but to keep it brief, these parasites might keep pest host populations in check. They might lubricate vital predator-prey interactions, and they might actually be responsible for subsidizing apex predators. And losing these parasites could mean losing the ecosystem services that they provide. Meanwhile, the winners are probably going to include some parasites that we do not want around, pests of aquaculture and wildlife, and potentially pests of people. We've learned a lot from studying marine disease in contemporary ecosystems, but now that we have a way to get into Jurassic Park, I am not hopping on a helicopter and leaving. We have an opportunity here to resurrect long dead information that most people thought was gone forever. And this information could help prepare us for a changing world. Thank you.
Fantastic talk. Thank you so much. It's really a fascinating topic. And I was wondering, because you were saying that there are cases where fishing interrupts the life cycle of trophic parasites, trophically transmitted parasites, but there can also be cases where parasite abundance increases because of increased host population density. So I was wondering, what's your take on whether conservation can impede or support infectious disease control, or is it more of a case-by-case scenario where you have to look at each population or species or parasite type in part?
Yeah, I mean, I think I wish that I had some like grand generality to offer, but it really does look like there's it will be a case-by-case consideration. I think about this a lot in reference to human disease, but let's think about it in the context of like coral reef parasites, since that's what this talk primarily focused on. We saw in the coral reef data, I'll scroll back to that for a moment so that we have that plot handy, that the directly transmitted parasites tended to increase, and that was because the fish that we were able to have access to were primarily small, low trophic level fish, right? Like it's really hard to get a scientific collecting permit for sharks in a completely undisturbed coral reef ecosystem. So this response for the directly transmitted parasites could have looked a lot different if we were able to sample some of the hosts that were declining in abundance, because we would expect that for the hosts that are diminishing in density due to fishing pressure, we would see a corresponding decline in the directly transmitted parasites associated with those hosts. That's not reflected in this data set because we weren't able to sample those high trophic level hosts. We were only able to sample low trophic level hosts. So I think the one generality that we can offer is that if you're looking at fishing specifically, it does seem to be really consistent that directly transmitted parasites track their hosts and trophically transmitted parasites just go kaput in the presence of fishing. The complicated thing about looking at change through time is that fishing is only one part of what's happening. There are lots of other changes happening in that ecosystem. And the history of fishing isn't all that simple, particularly in developed countries where we now have fisheries regulations and conservation laws that protect these populations. It's especially for English sole. English sole was really heavily fished until the 1940s. And since then, there's been a relaxation of that fishing pressure, partially due to closure of the English sole fishery, and partially due to conservation legislation implemented to protect other groundfish like rockfishes. So, English sole density has not nosedived over the period of our study. It's actually gone down really strongly from the 1940s for another decade or so, and then it's increased again. So we really need to look not necessarily at time as our driver, but at host density to try and understand what the influence of fishing is relative to the many other things that are changing.
Very interesting. There's a question about the sampling from the museum collections. So how does sampling affect the fish samples? And how did you manage the sampling? Did you sample from collections that had above a certain lot size? And also, do you think it's possible that your sampling is going to incorporate issues related to population densities or local abundance?
Yeah, so when we started this museum specimen stuff, I expected to have to really convince museum curators to let us examine their specimens, in part because our dissections are partially destructive. So we will take a fish, open up its body cavity, and pull out all of the viscera, as well as one of the gill arches and the heart. And we then basically destroy those viscera. We put them back into the lot from which they came and make sure that those the remains of the viscera can always be associated with their original host, but the viscera are destroyed. We usually, when we're dissecting fresh specimens, will take a fillet and squash that fillet between glass plates and throw a strong light through it and examine it under a microscope to look for any encysted parasites in the musculature. But with the museum specimens, we skip that step because we don't want to be that destructive. And instead what we do is we pull the body cavity open and shine a strong light through either side of the fish so that we can find any encysted parasites, basically through candling. So we have this kind of modified dissection protocol that is still destructive, but leaves most of the external morphology of the fish intact. And we've worked really closely with museum curators to make sure that what we're doing to the fish is as minimally destructive as possible for the information that we want to get out of it. And so far it's been great. The curators have been really supportive, and they're excited to have science going on in their collections because that's how they justify continued funding, right? They want to show that papers and insights are coming out of the collection so that the powers that be are convinced of the value of spending millions of dollars to maintain what is essentially a dusty old basement full of dead fish that no one ever visits. So we work with the curators on the actual protocol. We also work with the curators on which fish to choose and how many we're allowed to take. And we only select host species that are really well represented in the museum so that we're only dissecting a small fraction of the total holdings of a particular species from a particular place. And we actually have an approved list of lots before we start any dissections. So that's an agreement that we make between us and the curators. And then we also try and kind of distribute our dissections across multiple museums in cases of hosts that aren't super well represented. And we've been working with a number of folks, including the Scripps Ichthyology Collection, the Cal Academy, Smithsonian, National Museum of Natural History, American Museum of Natural History, and they send us specimens as well to kind of fill in some of the timeline gaps that we have from just looking at the Burke specimens. And then as for the last part of your question was about population density, that is not something that we can get at from the museum specimens. Maybe there's some fancy geneticist out there who can pull good genetic information from formalin fixed specimens, which is something that is moving forward, not in my lab, but in the labs of collaborators. But what we can do is collaborate with people who are interested in the historical ecology of fisheries to share data on fish density over time. And that's what we're doing with Tim Essington, Jamil Samhori, and Corey Green. They've all got a long time series of fish density, or at least fish relative density from catch per unit effort. over many decades, and they're sharing those data with us so that we can look to see whether fish density is a predictor of parasite abundance in ways that are not shown in the correlation with time.
Cool. Were you able to detect changes in host responses to parasite over time? And this is in reference specifically to Daniel Bolnick's recent work showing that fish immune responses to tapeworms can evolve rapidly, which they find partially by looking for tapeworms that grow to much smaller within host sizes.
Oh, interesting. So we haven't been measuring the parasites. What we have been doing is vouchering all of them. So every parasite that we find gets pulled out of the museum specimen fish and put into its own voucher file. And they're all going to be lodged in the Manter parasitology collection at the University of Nebraska, but currently they're all in our lab. So we could actually measure the parasites and test that hypothesis. We haven't noticed anything yet in the historical collections, but in the coral reef study, we did notice that the grouper that we worked with has this really strong immune response to parasitic infection, where it like covers the parasite with this black melanized tissue. And you can see that for a number of parasites that occur in the groupers, and it's common across the family. But one thing that we noticed was that the groupers appeared to have a much more effective melanization response to nematode parasites, encysted larval nematode parasites, on the unfished island than they did on the fished island. So we found way more like dead husks of nematodes inside this melanized material on the unfished islands than we did on the fished islands, where we tended to find live nematodes. There are a lot of potential explanations for that, but one is that the fish on the unfished island are better at mounting an immune response than the fish on the fished island. We haven't followed up on that, but we have the data if anyone's interested in pursuing it.
There's another question on our Slack channel. Do we know anything about how these parasite species vary in terms of how host specialists versus generalists they are? And if so, have you looked at whether this leads to differences in parasite responses over time?
That's such a good question. I wish that we had the information that you describe on host specialization. Most of the parasites that we encountered in the coral reef project are probably undescribed. So we identified them to the lowest possible taxonomic level, but we're not parasite taxonomists. So we couldn't describe the 77 new species that we detected in that project. Because it's such a remote location and an unstudied one, we just don't have the taxonomic effort going into describing that particular parasite fauna. And because we don't have them described, we can't say with certainty what their life cycle is and what hosts they're limited to. So in the Coral Reef Project, we did try to get at this just by making assumptions about the host specialization of the larger taxonomic group that each parasite belonged to. So for example, monogenes have a reputation for being really, really host specific. And things like gnathiids have a reputation for being really, really generalist. So we assigned this like ordinal score for host specificity to each of the parasites on the basis of its membership in that larger taxonomic group. And we looked to see whether that was related to their response to fishing pressure, and it wasn't. But it's not a very strong test because we don't know for sure about the host specificity of the particular parasite of interest. We just made assumptions based on our belief that host specificity would be conserved phylogenetically. So it's an unanswered question, one I really wish we could get at.
Cool. I think this is all the time we have for today, but thank you so much, Chelsea, for accepting our invitation and for your excellent talk.
Thank you guys so much for having me, and thank you for your patience with all of my copyright violations. I think the YouTube police are here to drag me away now, but it was a pleasure.
That's great. For everyone watching, do join us again on Wednesday for another very exciting talk. Dr. Susan Johnson from the University of Edinburgh will talk about the evolution of individual recombination rates in the wild, so don't miss it. Until then, have a great day, everyone. Stay safe and goodbye.